Okay, it's great to be here, and I want to thank the organizers for giving us an opportunity to present. Our work comes from the splicing field, and we are new to autism, and so it's really great to hear all the fascinating talks. And so our lab is at the medical faculty at the Hebrew University, and we are in the same campus as the Hadassah hospital. And this story starts there with two parents that are first cousins, and their firstborn, who was diagnosed with developmental delay, intellectual disability, and autism. And after exome sequencing, it was found that it has a homozygous mutation at the Wp4 gene. And so this allowed the parents to then undergo genetic counseling, and they now have two healthy kids. They did have a few miscarriages along the way, and it was attributed to the fact that the woman has a marker chromosome. But the last abortion that they had, they actually did exome sequencing and found that it was homozygous to Wp4. And so this kid has many regions in his genome that are homozygous, right? Because his parents are first cousins. So then to really convince you that it is Wp4, we now have two other cases in the world, one in Estonia and the other in Paris, that have kids that were also born to first cousins. And they have, in a way, a similar phenotype, and they have a developmental delay. One of the kids died when he was a year old, so we don't really have a full phenotype, but it died because of something that was not had to do with his neurodevelopmental delay, but rather an infection. So it does seem like it's Wp4, so now we need to ask, what does Wp4 do? And so Wp4 is a subunit of the spliceosome, and so 97% of our genes harbor introns, and introns have to be removed to allow for exons to ligate, and then for the mRNA to leave the nucleus and translate into a protein. And Wp4 is really one of the subunits of the core spliceosome. So the spliceosome is a really big complex that has 5 small non-coding RNAs and tens of proteins. And so a really big complex that does something that we need in every cell in our body. But this kid actually has only a phenotype that is associated with the brain. How is that? And so a glimpse to that we can get from the literature, and we know that some of autistic individuals actually have different splicing patterns in their brain. And I think that the most strong connection of splicing and autism actually comes from the fact that 10 of the genes that we know that are associated with autism are actually splicing factors. And so they are not part of the core spliceosome, but rather proteins that regulate the spliceosome. And so they show to cause a change, of course, in splicing and also autism. And another thing that we know, that the biggest group of genes that is associated with autism is actually chromatin genes. And chromatin is an intimate regulator of splicing. And so this, and of course, changes in chromatin that are associated with autism were shown to change splicing. And lastly, environment, we know that drugs that women take during pregnancy can cause autism, and those drugs are sometimes associated with splicing. And so with that in mind, with this connection that can definitely be between splicing and autism, we went and asked, what does Wp4 do that causes this phenotype? And so I'll start by telling you really in a nutshell what Wp4 does. And so the way the spliceosome works is that it identifies sequences in the introns that are close to exons. And so we call them splice sites. So there's a complex that binds one splice site and another that binds the other. And what Wp4 do is that it bridges those complexes. And so it allows them to then get closer and for the exons to ligate to allow for splicing to happen. But actually, I told you that the spliceosome is tens of proteins. And so it's not only WP4, there are many, many proteins. And the way the spliceosome works is that everything is checks and balances. So there's one protein that binds, and then another binds to it and makes the binding even stronger. And everything is needed. And so what would you expect if I'll just take one off? How would that change splicing? And again, why does splicing only changes in the brain to cause pathology? And so with that, we started our investigation, and we started by taking fibroblasts from our mother, father, and proband, and doing RNA-seq, which I'm going to show you the results in a minute. But first, this proband has a homozygous mutation of a frame shift that is predicted not to give rise to a protein, and so we made sure of that. We looked at the WP4 protein, and essentially we couldn't find it in this proband. And so we went on and looked at the results. We looked at gene expression. How does gene expression changes? And we're expecting to see that it does change, right? It's different between you and I. It's different between men and women, different between kids and their parents. And so we were expecting to see a change. But in order to understand if this change gave rise to the phenotype that we're seeing, we asked, what are those genes that change? And so in the parent-specific genes, we couldn't see anything that can cause a disease. But in the kid, we actually could see stuff that are linking to skin, right? Because we were looking at fibroblast, and we can all appreciate that skin in kids is very different than adults, right? And so this is what we found. And it actually suggests that expression when you don't have WP4 doesn't change to cause the phenotype that we are seeing. So then we moved on and looked at splicing. And again, we saw a big difference. We were expecting that. And again, checking what are the genes that are changing. And the parent-specific genes didn't look to cause any type of phenotype, but we could see a phenotype in the alternative splicing events in this proband, and they were somehow linking to the nervous system. And so RNA-seq is very strong, but the analysis of the RNA-seq to try and see how splicing changes is very challenging. If you use different tools, they're going to give you different lists of isoforms. And so we wanted to validate our results. We wanted to use another method. We used real-time PCR to see that indeed the isoforms that we think we look at are indeed changing. And so to choose the events that we want to try, we looked at genes that were changing in splicing in the proband and were also associated with autism. And so from that list, we picked 11 genes, and I'm going to show you one of them today, LZTR1. You can see that most of our reads, this is RNA-seq results, most of our reads, maybe you can just see my note. Most of the reads, they align to exons. That's usually how we see RNA-seq results. But you can see some of the reads right in the middle that align to an intron. And some of the reads line to an intron in the father, also in the mother, and in the proband. But our RNA-seq analysis tool actually told us that there's more intron retention in the proband than his parents. And we wanted to validate that. And I want to say something about intron retention. Intron retention is an event of alternative splicing. When introns are retained, then the mRNA doesn't leave the nucleus. It's tethered. It's going to stay in the nucleus. But even if it's going to leave, it's not going to translate to a protein, because for the most part, you'll find a stop codon inside the intron. And so here's splicing that can regulate gene expression. Okay, so that's a way that it happens. But in this kid, it seems that intron retention happens even more. And somehow it makes sense if it has one subunit less of the spliceosome, when maybe splicing do happen less. And so to validate it, we looked at our fibroblast, and indeed we could see a change that in this proband, intron retention of this gene is higher than either of his parents. And the other 11 events that we checked, 8 of them really seem to be correct, and they validated. But now you want to ask, maybe it's not WP4. Maybe it's just the fact that he's very young and his parents are adults. And so to be sure that it is WP4, we have expressed WP4 in the cells. And so we either expressed an empty vector or WP4 that was conjugated to GFP. And so you can see from our results that when we stably transfect cells with empty GFP, we still see a difference between the proband and his parents. But once we rescue the cells, once we put in WP4, we lose this pattern of splicing. And so this actually suggests that splicing is different in the proband. But now you want to ask, how is it relevant? Because this kid doesn't have a phenotype in his skin. He only has a phenotype in the brain. And so, and splicing is very different. It's very different in the brain than any other tissue in our body. We have splicing factors that only express in the brain. We have axons that are only expressed in the brain. And so it's really essential to try and look in cells that are more relevant. And so in order to do that, we are in the process of taking our fibroblasts and reprogram them to make iPS cells and then differentiate them into neurons. We are somehow concerned with the fact that our cells have different backgrounds, even though they're one family and the parents are first cousins. We also want to start with a clean background, and so we're taking ES cells and using CRISPR to make isogenic cell lines with the hopes to try and make organoids and try to do RNA-seq either in the neurons and in the organoids to try and ask how is splicing different, and how is splicing different in cells that are more relevant to the phenotype that we are looking at. And another thing that we are doing is that we're using a unit that is based in our campus, and they're made a mouse model for us. You can see in green the guide RNA that we used, and we have one single frame shift, an insertion of one nucleotide in a mouse, and that actually recapitulates the mutation that the kid has. And once we'll have enough mice to work with, we hope to check splicing in the brain, to do RNA-seq, and to also check the mice behavior to see if we can see that they have autistic characteristics. And one of the things I didn't tell you before, but in the splicing factors that are mutated in autism, sometimes the patients miss the corpus callosum, the part in the brain that actually connects the two hemispheres. And the two other patients that we have in Estonia and Paris, they also are missing the corpus callosum. So we're really interested to see whether our mice, how does their brain look like. And so let me summarize what I've shown you, and it's that when you're missing WP4, that is a subunit of the spliceosome, splicing is different. We get more intron retention. It actually means that we get less splicing or less efficient splicing, and that the splicing in those genes that their introns is retained can somehow lead to the phenotype that we're seeing of this kid. This is very exciting because nowadays RNA therapy is a big thing. We have very small RNA sequences, RNA oligos, that we can insert into the brain and change splicing to cure disease. There's an FDA-approved drug that does this. And so trying to understand how splicing changes to cause autism can be very beneficial to us to try and change it. It's kind of like what we heard before, not really trying to change traffic because of what caused the traffic, but trying to find another road to take. And so this is what we think about our efforts. And so I want to finish by thanking both Eden Engel and Ophir Gaminder, who are MD-PhD students who are working on this project, and of course everyone else in the lab and our funding, and I'm happy to take questions.
Thank you.